Morning, welcome back. In today's lecture, we are going to talk about genome organization. In our last lecture, we talked about how information flow happens in biological systems. Gene is the basic biological unit of heredity. At individual organism level, the set of information in an organism is called the genome. So genome provide information required for function and survival of the organism. But when it comes to population, a population will carry large number of individuals, and each individual will carry set of genes and alleles. This set of information we call the gene pool. Now, by now, you have a clear idea what is a gene, what is the structure of a gene, and how it functions. You have learned this in your A-levels and also during your cell biology course. So, this lecture focuses on genomes, how genomes are organized to achieve maximum functional efficiency. Would it be possible to design a minimal genome? Is everything determined by genes? If so, it should be possible to design a minimal genome. Life can be distinguished from non-life when the entity can metabolize, respond to stimuli, or adapt and reproduce. If an entity is capable of doing all these, then there is life. So, can we identify the minimal set of genes required in such an entity? Mycoplasma genitalium has the smallest genomes of all free living organisms. So that is the closest we have to a minimal genome, naturally. But scientists were able to synthesize a minimal genome based on Mycoplasma genitalium in a test tube. This minimal genome, the synthetic minimal genome, carries 473 genes. Out of these 473 genes, large amount represent essential gene, but still there were some quasi-essential genes. These quasi-essential genes are not essential for life, but in the absence of these genes, the ability, the growth ability, reproductive ability of the organism is reduced. So, at least we know that the minimal set of genes is not more than 473. Compared to the minimal genome we just discussed, let us look at the viral genome. Virus, like many other parasitic organisms, have very reduced genomes because most of the biological functions required for their survival are done by the host. So they do not need these genes. Virus genome can be RNA or DNA, and there is such a big diversity in the structures. As you can see in this diagram, there can be linear or circular DNA molecules, linear or circular RNA molecules, and these molecules can be single-stranded or double-stranded. Regardless of this diversity in the genome structure, viruses carry few genes. For example, look at the genome organization of SARS-CoV virus. This group has genomes of 26 to 32 kilobases that encodes for more or less 9,600 amino acids. Altogether, there are 6 to 12 or 6 to 11 protein coding frames. If you go further down in this line, you would find viroids, which are circular RNA molecules, or even prions, which are simply protein molecules. These are not living organisms, these are particles, but still they are capable of transmitting genetic information from generation to generation. And their genomes carry sufficient information to complete the life cycle. So how you can apply central dogma to explain the behavior and the way information is transmitted in these particles. As an adaptation to their lifestyle, parasites like virus, phytoplasma, mycoplasma have reduced genomes. But if you take any free living organism, compared to the minimal genome, they have more complex genomes. You find bacteria at the lowest end of the scale. Bacterial genome consists of two components. We call them the chromosomal component and the extrachromosomal component. Chromosomal component, actually, although we call it a chromosome, it is not a true chromosome. It is DNA associated with some proteins. Usually, the chromosomal component consists of a single covalently bonded circular DNA molecule. But sometimes there can be multiple circular DNA molecules or linear DNA molecules. These DNA molecules are associated with some histone-like proteins. They are not really histones, but they're functionally equivalent. The extrachromosomal element, also called plasmids, are small circular or linear DNA molecules. These exist independently of the chromosomal element. The plasmids replicate independently of the chromosomal element. The plasmids carry genes that are not essential for the survival of the bacterium. However, these plasmids provide the bacterium some growth advantage. For example, some of these plasmids carry antibiotic resistant genes or genes that enable the bacterium to digest or metabolize specific substrate and so on. To get an idea about the bacterial genome, let's look at the genome of Escherichia coli, E. coli. E. coli chromosome is around 4,600 kilobases and around 4,300 protein coding genes were predicted in the E. coli chromosome. Out of these, the function is known for 1,800 genes. So that represent only 70% of the chromosomes. So 30% of the chromosome, the function of 30% of the chromosome is not known, even for a small organism like E. coli. However, compare this number with the viral genome and the minimal genome. If you stretch the E. coli chromosome to its maximum possible extension, it would be around 9 micrometers. But you know, bacterium, the E. coli bacterium cell is around 1 micrometer. So how this huge DNA molecule is fit into the small bacterium? This phenomenon is called DNA packing. Now you have learned bacterial DNA packing in your A levels, so I'm not going to discuss about it in detail. However, several proteins are involved, like topoisomerase and gyrases. These proteins fold the DNA into a very tight structure, and the proteins and the associated the DNA and the associated proteins make a gel-like, thick, viscous entity which we call the nuclei. Now let's look at the eukaryote genome. In eukaryotes, the genome is one or more linear DNA molecules. These are associated with histone proteins to form chromosomes. If you look at the human composition of the human genome, now that human genome is fully sequenced, only 1.5% of the total human genome represent coding regions. Coding regions means protein and RNA coding genes and also pseudogenes. Pseudogenes means genes that are no longer functional because of some defects. So both genes and pseudogenes accounts for only 1.5% of the genome. So large majority of it, more than 98% of it is non-coding regions. What these non-coding regions consist of? One important element is regulatory elements. Regulatory elements means DNA sequences that regulate, control gene expression. Very large fraction of the non-coding DNA is repetitive elements. We'll look at what they are. But then there are other DNA sequences in which the function is not known. These DNA sequences are also called junk DNA. However, not knowing the function does not mean that they are non-functional. Because during evolution, it's very unlikely an element that has no use is transmitted from generation to generation. You know how strict and how severe natural selection can be. Therefore, it's very likely that these junk DNA have some function, however, still we do not know. Compared to virus and bacterium, eukaryotes have big genomes. For example, the SARS virus had 11 genes, E. coli had 4,300 genes, whereas humans have 30,000 genes. Is there a relationship between the size of the genome and the complexity. If you look at these diagrams, you would see that there is no such relationship. The complexity of the organism is not correlated with the size of the genome. Not only size, complexity of the organism is not correlated with the number of chromosomes. Complexity of the organism is not correlated with the number of genes. These are paradoxes that we still cannot explain. Let's look at the eukaryotic genome in a little bit more detail. And here again we have the human genome. 1.5% of the human genome is coding sequences. But if you look at the non-coding sequences, some of them are associated with genes. We already said some of these non-coding regions are pseudogenes. Other than pseudogenes, you find some non-coding regions associated with genes. Eukaryotic genes, we know there are the 3' untranslated region and the 5' untranslated region. In 5' untranslated region, you find the regulatory elements, right? The promoter and enhancers and so on. In the 3' untranslated region also you find some downstream regulatory elements. Within the genes you find introns, which are also non-coding regions. Other than these non-coding regions that are associated with genes, there are intergenic sequences, that is sequences between the genes. What these intergenic sequences largely consists of, mostly they are repetitive elements. And then other than that, there are some junk DNA. Repeated DNA is a significant feature in eukaryotic genomes. Repeated DNA can occur in coding sequences as well as in non-coding sequences. In coding sequences, these are actually genes that occur in multiple copies. So all these copies together is identified as a family, gene family, because individual copies have more or less similar sequences. Sometimes individual copies can undergo differentiation, modification. However, on average, they have a more or less similar sequence, and also they are functionally related. So they exist as a family, and individuals in this family can be dispersed. So there can be intergenic sequences between these genes. In that case, the gene family is dispersed gene family. In other times, the copies can occur one after the other as a tandem array, so that we have a tandem gene family. Some of the repeats are non-functional, or we do not know the function of these repeats. There are large repeats in the centromere and telomeric regions of the chromosomes. These repeats are of hundreds and thousands of kilobases. And in contrast to that, there are tandem repeats, variable number tandem repeats, which are dispersed within the genome but occur as tandem arrays. These repeats can be 2 to 3 base pairs to 100 base pairs in size. One important and very interesting type of repeats are transposed elements. Transposed elements are also called jumping genes because these are mobile elements that can move from one place to another. Depending on the mechanisms they use to move from one place to another, there are two types of transposons. One is called actually transposons, the other one retrotransposons. Retrotransposons involve reverse transcriptase during the mobilization. Just like in bacteria, packing is very important in eukaryotic genomes. There is a systematic folding of the chromosome into primary, secondary and tertiary structures so that it can be fit into the cell. Now, DNA packing in eukaryotes, or you have learned this in your A-level, so I'm not going to discuss about it in detail. However, you need to know the processes involved. With this discussion on genome organization from virus to prokaryotes, the bacteria to eukaryotes, you would probably say it is not only the genes that matter. The genes carry the physical information, but the organization is also important. You can find a good analogy between a eukaryotic chromosome and a library. In a library, these books are arranged in such a way so that the clients, multiple clients, can access the books without damaging the books and at this very efficiently. That is the purpose of a library. Very similar thing happens in a cell. In a cell, the information in genes is accessed multiple times under multiple circumstances. And how the cell makes sure this is possible. Now you would probably be able to see the importance of the so-called non-functional DNA. What does it do there? It makes the shelves, the racks, the cupboards for the coding sequences. The coding sequences, as you learned in human genome, it is just 1.5%. The rest of it is just the infrastructure to make sure that these coding sequences are safe and accessible and the information is accurate.